Good afternoon, everyone. I'm Rishabh, and today we'll be starting the discussion week 12 of the course Experimental Biotechnology. And in this week, we'll be seeing the concepts that is basically the summary of all the things that we have studied in the course. Okay, so this is the last lecture of the course, and in this week, we'll be studying all the concepts and the questions related to that. Okay, so let's start. So this is the first question. The Organization for Economic Cooperation and Development (OECD) published the guidelines for testing the chemicals in the year dash that were internationally accepted. So what was the year in which it was internationally accepted? What can be the correct answer? So the correct answer is option number one. So in the year 1981, the guidelines that we have set up for OECD were published for testing the chemicals and they were internationally accepted. Okay. So it's a fact-based question. So the year was 1981. Okay. So let's move on to the next question. The recommended biosafety level for Lyme disease is, what can be the correct answer? So the correct answer is option number two, that is BSL2. Okay. Why BSL2? Because as per the different biosafety levels, the BSL1 are the organisms that are least dangerous to the environment. Okay. So least dangerous. For BSL2, we have those uh, organisms that have some dangerous or causes some. Okay, for BSL3, we have those organisms that causes serious infections in humans, plants or animals. Okay, and the last one BSL4, in which very serious infections are caused in humans, plants and animals. Okay, so these are the four BSLs that we are studying. For Lyme disease is a vector-borne disease. Okay, so it is caused by the hmm, so it is The actual organism that is causing the Lyme disease is bacteria, that is Borrelia burgdorferi. Okay, and it is transmitted. The vector is black tick. So it generally do not is it not it is not infectious because the bacteria doesn't transmit itself. It requires a vector to get transmitted. Okay, so it can be studied in a BSL-2 laboratory. If it would have been transmitted through their own, then it would be started in a BSL-3 laboratory. Okay, so this is how we'll describe the disease, that it is caused by Borrelia burgdorferi, and it will be treated in a BSL-2, Biosafety Level 2 environment. Okay, so if you guys have any doubt, you can ask. Okay, if not, then we'll move on to the next question. So, what is the pH of running buffer in SDS page? What can be the correct answer? So, the correct answer is option number 3, that is 8.3. So, whenever we are running a SDS page, so we have this like of situation where we have two. This is the stacking gel and this is the resolving gel. Okay, so the pH of the stacking gel is around 6.8 and the pH of the resolving gel is around 8.8, and the buffer that we use, that pH is around 8.3, so that it can basically make the different layer, the sandwich between the protein, glycine, and chloride ion. So this basically helps in movement of these three ions, this pH. So the correct answer is option number 3, that is 8.3. Okay, so do you guys have any doubt? If not, then we'll move on to the next question. Which of the following is a strong anion exchanger? What can be the correct answer? Sulfonic acid, carboxylic acid, quaternary amine, or diethyl aminoethyl cellulose? So what we generally see is, because the name itself is anion exchanger, so the bead that would have been attached would be positively charged. Okay, so anything like sulfonic acid or carboxylic acid, that would contain a carboxylic group here, so that would be negatively charged. So these two options are out of the context. Then we have two positively charged beads, that is quaternary amine and DEAE cellulose, so diethyl aminoethyl. So as you know that quaternary amine means that you have a carbon group that is connected to three nitrogens. So this is a tertiary and one more, that is a quaternary amine. Okay, so in terms of the structure, and diethyl aminoethyl is basically uh, you have two ethyl groups, sorry, yeah, two ethyl groups. Just wait a second. So it's like this. So the cellulose is here, and from this it is connected to ethyl, then amine group, then again two ethyl molecules. Okay, so what happens in this case, because this is a charge donating group in terms of its nature. Okay, so it basically donates the group, so it decreases the amount of positive charge on the nitrogen. Okay, so that's why this is not a strong positive charge. Okay, but in this case what happens, when there are many nitrogens, so more nitrogens try to snatch electrons from the carbon. So in that case the other two nitrogens become more positively charged as compared to the other two. Okay, so in this case what happens, these nitrogens are more positively charged, so they can hold very hold the negative charge strongly. Okay, so that is why these are the strong anion exchanger. Okay, so this is the correct answer. So quaternary amine is a strong cation exchange. Okay, so any doubts? Okay, if not, then we'll move on to the next question. The proteins have minimal solubility at, what can be the correct answer? So the correct answer is, because here Pi stands for the isoelectric point. And remember that whenever the pH is near to their isoelectric point, the protein have minimal solubility. Okay, so the correct answer is option number one, and here is the explanation. Okay, so whenever we have the pH that is less than Pi, then the protein would be positively charged. Okay, and the vice versa case, when pH is greater than Pi, then the protein will be negatively charged. Okay, so now what happens whenever the pH is near to the Pi? So the net charge is zero. That means there are equal groups of both positive and negative charge on the proteins. So what happens? This basically helps the protein to get detached from the interactions of the water or any solution that it is in. So what happens generally, the solution would have both the negative as well as the positive charges. So now if that is equal in that case in a protein, so what happens, the interaction between the water molecule and the protein molecule basically hinders up, and that's why it helps the protein to get precipitated, and it has minimal solubility in the solution form. Okay, so this is why the pH, when it's becoming equal to the pi, that means at that point of time we can precipitate the protein and can be used for further analysis. And if the pH is really less than or greater than the pi, in that case it will get dissolved in the solution, because the because of the positive and the net negative charge that basically stabilizes the protein in the solution form. Okay, so that is why the minimal solubility is observed in the pH in the protein that is near to the pH. Okay, so any doubts? Okay, if not, then we'll move on to the next question. In affinity chromatography, which of the following has affinity towards lysine as a receptor? What can be the correct answer? So what happens, out of all the proteins, there is only one rRNA. So in literature, it suggests that because rRNA has a net negative charge because of the phosphate group present, because it's a nucleic acid, so it has a net negative charge. So for the separation of rRNA, there is a possible it has been seen that it is attracted as a lysine receptor towards the lysine receptor. So whenever we have to purify rRNA, we can run a column that have a lysine at its base so that it can be separated easily. Okay, so this is the case. So rRNA is the correct answer. Answer option number 4. Okay, so any doubts? Okay, if not, then we'll move on to the next question. Complete Freudent's adjuvant of heat killed. Well, which organism is present in complete fraudulence adjuvant? So the complete fraudulence adjuvant contains the heat-killed spores of Mycobacterium tuberculosis. So the correct answer is option number 2. So basically what happens, why we need this? This is basically a immune stimulant. Okay, so this immune stimulant basically helps in uh, starting the immune response. Okay, so that is why the adjuvant contains the spores of the Mycobacterium tuberculosis. And in case of the incomplete adjuvant, it's the oil emulsion. This is for incomplete adjuvant. Okay, so this is the case. Let's move on to the next question. The phenomena of clumping of RBCs by the viral particles is an example of precipitation, immunoprecipitation, agglutination, or immunodiffusion. What can be the correct answer? So the correct answer is option number 3, and here is the explanation. So whenever we uh, do see a clumping of RBCs with the chiral particles, so what we see is this type of clump is formed. So this is basically agglutination of the RBC. Okay, and the this leads to RBC lysis basically. Okay, and whenever we have to reverse this, so what we do, we use an antibody against the virus. So in that case, what will happen, the virus will not be able to aggregate the RBCs. So what will happen, the antibody basically inhibits the virus to make an aggregate with the RBCs, and the RBCs will be free. So this inhibits the process of the agglutination in case of RBCs. Okay, so let's move on to the next question. Trypan, the this is a assertion reason type of question. So assertion is trypan blue stains only the dead cells, and the reason is trypan blue is a charged dye and cannot enter the live cells due to the presence of the membrane potential. So here are the options. What can be the correct answer? So the correct answer is option number 2. That is both assertion and reason are 2, and the reason is the correct explanation for A. And here is the explanation. So whenever we use a trypan blue, so it basically helps us to check whether the cells are viable or not viable. Okay, so what will happen? The trypan blue is a negatively charged type, and as we know that the cell surface is also negatively charged, so the membrane potential makes it a negatively charged. So whenever cell will be alive, then the negatively charged will be there, and so what will happen? It will not be able to enter the cells. But whenever the cells are dying, so in that case what will happen, there is a change in the membrane potential. So the trypan blue can enter the cell now and can stain the and can accumulate in the cytoplasm. So the dying membrane becomes permeable to the trypan blue and gets accumulated in the cytoplasm, thus appearing blue when we see in the microscope. So this is a dead cell and this is a live cell. Okay, so the membrane potential of the cell basically prevents trypan blue from entering the live cell, and it allows the cell to enter into the dead cell. Okay, because of the change in membrane potential. So this was the correct answer, the option number 2. Okay, so let's move on to the next question. Which of the following cells has the smallest diameter? The options are RBCs, lymphocytes, macrophages, or megakaryocytes. What can be the correct answer? So the correct answer is RBCs. Okay, and it has the smallest diameter, as we can see from this. So the smallest is the platelets. So platelets are basically derived from large cells. These are megakaryocytes. Okay, and this has the largest size as compared to the all other cells. And then comes the diameter of erythrocytes that has the RBCs. Okay, then comes the lymphocytes. So after RBCs we have lymphocytes, and after that we have macrophages that are nothing but the monocytes, and then comes the megakaryocytes. So this is the correct order of the increasing order of diameter. So RBCs has the smallest, then lymphocytes, then macrophages, and then megakaryocytes. Okay, so let's move on to the next question. Fluorescein isothiocyanate on excitation emits, options are red fluorescence, blue fluorescence, orange or green fluorescence. What can be the correct answer? So FITC on basically exciting, it leads to green fluorescence on emission. Okay, so the correct answer is option number 4, and here is the explanation. So this is the excitation wavelength of the it is in the blue region. Okay, so it is between the 450 to between four nearly 500. So between 490 to 500, it's the wavelength of excitation, and the emission peaks lie in the green region, that is nearly around 525 nanometer. So this is the green wavelength that is being, so this is the case. Okay, so it emits green fluorescence when FITC is excited. Okay, so let's move on to the next question. Which of the following has the highest density? The organelle, protein, DNA, or mammalian cells? What can be the correct answer? So this question is based upon the amount of the particular molecule present in the cells. So in this case, DNA has the most amount inside the cell. Okay, so that's why it has the highest density. Okay, so the correct answer is option number 3. Okay, so let's move on to the next question. How much quantity of NaOH is required to prepare 1 liter of 0.025 normal solution of NaOH? What can be the correct answer? So here what we will do is, so NaOH, we consider it will donate only one OH ion. So okay, so in that case the normality and molarity of NaOH would be same. Okay, so this means that 0.25 molar of NaOH solution we have to prepare, that is 1 liter. Okay, so molarity is basically number of moles per liter. So 0.025 moles in 1 liter solution. We have to prepare 1 liter only. Okay, so now we have 0.025 moles of NaOH. So in that case, 0.025 moles gives the given weight upon the molecular weight. Okay, so we have to find out the given weight, and the molecular weight of NaOH is 40 gram. Okay, so the weight will be into 40, so that will come around 1 gram. Okay, because this will cancel out everything. Okay, so this will be 1 gram, that is option number 2. Okay, so let's move on to the next question. In phenol chloroform method of DNA extraction, the DNA is present in which part of the mixture? The options are aqueous phase, organic phase, interface between the aqueous phase and the organic phase, and or in the thin layer between and on top of the aqueous phase. What can be the correct answer? So what happens, given the nature of the DNA, it's negatively charged. Okay, so it cannot be remain in the organic phase. So it will be present in the aqueous phase only. Okay, so the correct answer is option number one, that is the aqueous phase. Okay, and here is the steps that we follow in case of DNA isolation from any organism. Here we have chosen the blood. So what we do first, we do in case of the blood, we do RBC lysis. That means only WBCs are present in the cell, in the solution from which we generally take the uh, DNA. Okay, then we do the SGS, EDTA, DTT, and proteinase K treatment. So what will they do? So basically, they have to denature the proteins, the cell membrane, different organelles that we have, and the nucleases that we have. Okay, so all these things will be deactivated and degenerated using all these proteins. Okay, SDS is a negatively charged detergent. EDTA is a chelator that basically chelates the ions so that the nucleus cannot act. DTT is a reducing agent, so the proteins can get denatured, and the proteinase K is also a protein denaturing agent that helps in denaturing the proteins. So these four uh, things will help in denaturing all these extra amount other than the DNA. Okay, and then we have a mixture of phenol, chloroform, and isoamyl alcohol that is in the ratio 25 is to 24 is to 1. Okay, so this is the ratio of all the three. And what will happen? The phenol and chloroform will form the organic phase, and isoamyl alcohol will help in formation of the um, will help in formation of the aqueous phase. So now when we add this particular mixture into the solution of DNA, so in that case we have two layers, that is the aqueous phase and the organic phase, that is the below one. So the organic phase would contain all the lipids because they cannot go in the aqueous phase, and the at the interface we have all the proteins, and on the in the aqueous phase we have DNA. Okay, so this is the major amount that is present in the aqueous phase, and we collect the aqueous phase in a different test tube, and then what we do, we either add isopropanol or ethanol for its precipitation. Further, the ethanol washes for precipitation. So this is the precipitated DNA that we have isolated from the particular cells. Okay, so this is the basic steps that we follow for the phenol chloroform method of DNA isolation. And the DNA would always be present in the aqueous phase. Okay, so any doubts? Okay, if not, then we move on to the next question. A research fellow is facing problem of acrylamide polymerization even when he is allowing polymerization reaction for 30 minutes at room temperature. Which of the following component is most likely to be the missing from the cocktail? The options are glycine, SDS, HCl, or TEMET. What can be the correct answer? So in this case, as you know, the TEMED basically helps in initiating the free radical mechanism. Okay, so that basically helps in polymerization. Okay, so the correct answer is option number 4, and here is the process of polymerization that basically takes place. So acrylamide and bisacrylamide are the monomers of the particular page gel, and TEMED basically starts the or basically stabilizes the free radical formation, and APS is basically the catalyst for free radical formation. So, if these two have a problem, then that would be not enough for the formation of the free radicals, and that would hamper the polymerization step. So, when kept in incubation with TEMED and APS, this needs to be taken in mind that both needs to be prepared afresh. Okay, so if both of them have some problem, then that would lead to the problem with the polymerization. Okay, so this is the case. So the correct answer is option number 4, that is TEMED. Okay, so any doubts? If not, then we'll move on to the next question. The KD values of four proteins are given below. But and if the mixture of these proteins is loaded on a gel filtration column, which protein will come out last from the column? What can be the correct answer? Okay. So, as we have discussed in the previous lectures, KD stands for the distribution coefficient, and it is generally inversely proportional to the molecular weight of the analyte that is needs to be separated from the gel filtration column. Okay. So whenever Kd would be high, the molecular weight would be less. So that means if the molecular weight is less, that is will be directly proportional to the retention time. Okay. So when Kd will be high, molecular weight will be less, and that would lead to more retention time. Okay. So if there is more retention time, that means it will remain for the more time in the gel filtration column. So in this case, if we see protein A as 0 kd, that means it has protein B has kd1, C has 1.5, and D has zero point 0.5. Okay, so that means the molecular weight of A would be the least. Sorry, molecular weight of A would be more, then comes point 0.5, then 1, and then 1.5. So for 1.5, the molecular weight would be least. That means it will have more retention time. Okay, so this option, the C protein, would be the protein that would travel the most distance inside the column, and it will remain for the most time in the uh, gel filtration column. So the correct option is option number 3, that is protein C. So it will remain for the more time in the column and will elute out last. Okay, so any doubts? Okay, if not, then we'll move on to the next question. The nuclear protein is involved in regulation of gene A. The protein was incubated with gene A and run in a gel filtration column. The chromatogram recorded. Uh, Select the correct statement from the following options. What can be the correct answer? So, what will happen uh, when we have the protein? So, for example, this is a gene A, and here is the protein that is interacting with the gene A. Okay, so if we run this particular column on the gel filtration column, so what will happen if we run only the protein, DNA, and protein plus DNA? Okay, so obviously the protein would be circular and it would be having a smaller molecular weight. So molecular weight or the hydrodynamic size that we talked about would be less. So it will have more retention time. Okay, same is the case with DNA also. But it would be a bit straight, So it can, because of the hydrodynamic size, it can elute earlier. But in case of the protein plus DNA, what will happen, because this would have more hydrodynamic size, so that would lead to more molecular weight and lesser retention time. Okay, now we'll see the options that we have. So the hydrodynamic surface area of the protein ligand complex is lesser. So no, it will be more, because the protein has already bound to the DNA. So that would lead to more molecular weight. Okay, that we have just now discussed. Okay, now, the peak corresponding to the protein ligand complex will show a right shift. No, that would be a left shift, because it would be eluting out first. So, what does the mean by the right shift? So, what happens whenever we graph it? So, based upon the absorbance and the molecular weight, we see a peaks. So, the initial peak would be for the protein that have highest molecular weight, followed by the lowest molecular weight. So it would not see a right shift, it would be a left shift. So the protein would be coming around somewhere around here only, and it would see a left shift whenever the protein is bound to the DNA. Okay, so the second option is also wrong. The Kd value of protein ligand complex will be more. No. Why? Because as we discussed, that Kd is inversely proportional to the molecular weight, and if molecular weight is more, then Kd would be less. Okay, so here in the case where protein is bound to the DNA, so it would have less Kd. So this option is also wrong. The protein ligand complex would have lesser retention time. So yes, it would have lesser retention time, because the highest molecular weight will elute out first. So the retention time inside the column would be less. So the correct answer is option number 4, that it has lesser retention time. Okay, so let's move on to the next question. A mixture of arginine, phenylalanine, and histidine was fractionated using cation exchange chromatography at neutral pH. The amino acids were separated with an increasing salt gradient. So we have to identify the correct order of elution. What can be the correct answer? Okay, so we are using a cation exchange chromatography. So that means the protein or the amino acids that we are seeing, that would have a positively charged group. So arginine is also a positively charged amino acid, and histidine also, and phenylalanine also. No, sorry, phenylalanine is a or aromatic amino acid. So at neutral pH, that is at 7, we'll consider. Okay, so arginine has a pi that is greater than 7. So whenever, at 7, that will be less than pi, so it will be positively charged. Okay. For histidine, the pi value is less than 7. It's around 6 point something. Okay. So 7 would be like near to its physiological pH. Okay. So that means it would have a near neutral, so it would be zero charge nearly. Okay. And in case of phenylalanine, because it's a aromatic amino acid, is pi lies near around 8. Okay. So it would be also positively charged, but the net positive charge would be less than the arginine value. Okay. So whenever we have to purify them, what will happen? Based upon the charge, they will be isolated. Okay. So first, because it's a net negative charge, histidine would be eluted first. Then comes the phenylalanine, and then comes the arginine. So this is the correct answer. So the correct answer is option number 2. Okay. So that means the phenylalanine will be eluted first, and then comes the histidine. Okay. So this would be the correct answer. Just a minute. Oh, PI of phenylalanine is around 5.76. Okay. And for histidine, it's 7.64. Okay. So 7.64. Okay, that was my mistake. So the that would have negative charge at neutral pH, and this would also have positive charge, but that would be less than arginine. So the correct order, that would be phenylalanine, and then histidine, and then it would be arginine. So yes, the correct answer is option number 2. Okay. So yes, the option number 2 is the correct answer. Okay. So let's move on to the next question. A purified oligomeric protein was analyzed by SGS page under reducing and non-reducing conditions. A 1 liter solution of 1 mg per ml concentration has 4.01 into 10 to the power 18 molecules of the oligomeric protein. Based on this data, deduce the total number of polypeptide chains that constitute this protein. So we are given 1 liter of 1 mg per ml concentration of the protein, and we have 4.01 into 10 to the power 18 molecules. Okay, so this would be, if we see that the in 1 ml 1 mg protein is present, that means for 1 liter 1 mg 2000 in 2000 ml. Okay, so this would be 10 to the power 6 mg of protein that we have. So so 75 kilodalton would be the molecular weight of the protein, because it's in non-reducing conditions. So this much molecules would be present in, or else we can go the other way around also. So in 4.01 into 10 to the power 8 molecules are present in 10 to the power 6 mg of protein. So the 6.02 into 10 to the power 23, this is basically the Na, that is Avogadro number. So this would yield the molecular weight of the oligomeric protein. So this would be 10 to the power 29, 10 to the power 29, and this would be 10 to the power 11 mg. Okay, so that means 1.5 into 10 to the power 8 gram. So here we would have 1 gram of protein, not 10 to the power 6. That was a calculation mistake. So in this case, what will happen? These many molecules are present in 1 gram of protein. So out of the Avogadro number, we would have around 1.5 into 10 to the power 5 uh, grams of the protein. That means this yields us the molecular weight of the protein based upon the number of molecules that are present. Okay, so in this case, what will happen? If we see that from this graph, the 75 kilodalton is the molecular weight that is coming from the for the oligomeric protein. So now this was the molecular weight that we are getting divided by 75,000 daltons. So basically, gram or Dalton conversion is not that big. So we can consider both as same. So now if we divide them, then this ratio comes out to be 2. Okay. So that means the protein is made up of two polypeptide chains. That means one is of 50 kilodalton and one is of 25 kilodalton. So the correct answer is option number 2. Okay. So let's move on to the next question. So the correct answer is option number 1. So this is wrong here. Okay, let's move on to the next question. Techniques that can be used to check protein-protein interactions that can be used are yeast to hybrid system, phase display, or density gradient centrifugation. What can be the correct answer? So the correct answer is option number 4, that is all of the above. So these are the new techniques that can also be used. So sorry, this would be option number 4. Okay. So these are the techniques. So first I'll explain briefly about the yeast to hybrid system. Okay. So what happens is, as we know, in case of a transcription factor, it would have two domains. First is the DNA binding domain, and the second one is the domain that connects it with the DNA polymerase. Sorry, RNA polymerase, so that the polymerization reaction can be taking place. Okay, so this is the DNA binding domain, and this is a protein binding domain. Okay, so now when both of them are here, that means, if we cut this transcription factor into two, and if we have to check whether two proteins are interacting with each other, and we are able to synthesize a artificial protein in which one part of the protein it is bound with the DNA binding domain, and the other part of the protein is binding with the protein binding domain. So what will happen if both the proteins are interacting with each other? This is the part where the DNA binding domain will lie, and here the protein 1 will be there, and it will interact with protein 2, and that have the protein binding domain. So if both the proteins are interacting, that means if we the polymerization will take place, and if both the proteins are not interacting, that means the polymerization will not take place. Okay, so this is the concept that have been used for the yeast to hybrid system. Okay, so the DNA binding domain is bound with the target protein, and the transcriptional activation domain or the protein binding domain have the binding partner. Okay, so if the both the proteins are interacting with each other, and this is a reporter gene. Okay, so this can be GFP or luciferase, what can be there. Okay, so that can be used to report the transcription analysis. Okay, so now the DNA binding domain would have the target protein, and this target protein is binding to the binding partner, and here is the protein binding domain, and this will help the RNA polymerase to follow the expression pattern. Okay, and this would produce the reporter protein. So this would allow in the in vivo system whether the two proteins are interacting or not. Okay, so this is the case with the yeast to hybrid system. Now, moving on to the second option, that is phage display. So, in that case, what happens? We produce some bacteriophages that are expressing a particular protein on their surface. So, now what we do is we have a plate kind of format that have certain proteins bind to it, and if one of the proteins that have been expressed on the surface of the phage binds to those proteins, we generally uh, separate out those proteins, those phages that have that particular protein, and then we separate out, and then we do their amplification. Based on that, we generally find out that this particular protein that has been bound to the surface of the phage is basically interacting with the proteins that are bound on the plate. Okay, so that helps in checking the binding patterns of the protein-protein interactions between two protein molecules. And third is the density gradient centrifugation. So as you can see from the Michaelis sorry, measles stand experiment of DNA. So they used a cesium chloride based centrifugation system to separate the high and the low radioactive N molecules that have been used. So between the N15 and N40. Okay, so based upon that, because this is sensitive, so we if the two proteins would be interacting, the if we separate out in a single um, centrifugation reaction, so we can see if both the proteins would be interacting, they would form a band that would be higher as compared to the band that of the different proteins, separate proteins. Okay, so this is how we can use the density gradient centrifugation also to check out whether two proteins are interacting or not. There are or other methods, so that these are the few methods that are recently developed. Even we can use FRET. Okay, so this is a fluorescence based technique in which the two proteins that would be interacting are bound to different fluorophores, and there would be a third fluorophore. Okay, so if we give the excitation wavelength of this particular fluorophore, and the emission wavelength of this fluorophore is observed, that means both the proteins are interacting, and they because these two fluorophores would be uh, nearer to each other, so that means the emission wavelength of this fluorophore can be observed. Okay, so this is also one of the techniques with which we can check with the protein protein interactions. Okay, so let's move on to the next question. Okay. So, in a confirmatory test for HIV, one or more viral antigens are detected in the blood of patients. Following are the steps to be performed for the experiment. So, there are these are the steps. So, the transfer of viral antigens to the nitrocellulose paper, incubation with the buffer containing antibodies specific for viral antigens, separation of viral antigen by SGS page, or detection of bands by enzyme linked secondary antibody. So, what can be the correct sequence of steps? So, first we have to purify the HIV protein or the HIV antigen. For that, first we'll do SGS page. Then we will transfer it to the nitrocellulose paper. That means Western blotting. Okay. Followed by this, we will have a antibody specific to the antigen. Then we would have the secondary antibody linked to the enzyme. Okay, so these would be the correct steps for the identification of the sorry, for the performance of the steps experiment. Okay, so the first step would be this, second would be this, third would be this, and fourth would be this. Okay, so the R would be the first step, then we would have P, then Q, and then S. So the correct option would be option number 3. Okay, so this leads to the end. Oh, there is one more question. A solution of a mixture of proteins is loaded onto a gel filtration column that has a size exclusion limit of 75,000 Daltons. The proteins have the following molecular mass in Daltons. So A have 11,000 Daltons, 66,000 Dalton is B, C is 42,000 Dalton, and D is 90,000 Dalton. The protein will elute in the from the column in the following order. So as we can see, D has the highest. So D will be the first one to elute. Then we have B, then we have C, and then we have A. So based upon in the gel filtration column, the size limit is this. So the first protein that will be eluted will be the highest molecular weight. So D will be eluted first, then comes B. This is the second highest. Then comes C, that is the third highest, and A that would remain in the column for some time and then it get eluted. So the correct answer would be option number 2, that is DBC. So this leads to the end of our discussion, and yes, thank you guys for joining. And if you have any doubt, you can reach out to me. Okay. So thank you for joining in.